All right, we're recording. Hello, everyone. Welcome to Skype a Scientist Live. My name is Sarah McAnulty. I am the Executive Director of Skype a Scientist and run these live sessions. We're joined today by Devin Swiner, a chemist. We're really excited to have Devin on. Tomorrow is Devin's defense date. So that is what happens at the end of your PhD. It's like your final test. And then after that, you're a doctor. So it is an exciting week in chemistry land for sure. We're also joined by Erin Fell, our ASL interpreter. So we're really excited to have both of you with us today. Housekeeping, there isn't any. Oh, on Wednesday, so this week, Wednesday, 1 p.m. Eastern, we're going to be talking with Jimmy Burnett all about the ocean and all the things that live within it, and specifically ocean parasites is one of the things that he knows a lot about. So get excited for that. It's gonna be good. But for now, let's get into it. Devin, thank you so much for being with us here today.
Thank you for having me.
Yeah, it was just great. It's what a week for you. So do you want to tell us who you are, what you do, and why you like it?
Yeah, so hi everybody. My name is Devin. I, like Sarah said, am a fifth year. Well, I guess I'm graduating tomorrow. That's how my advisor puts it, which is really weird to think about. But I am an analytical chemist. And I go to The Ohio State University. And I'm heading to pharma in a couple of months to start a job at Merck in June. So I'm going to show you all a couple of slides about how I use thread to do a lot of chemistry. So if you have any questions, I will really answer anything. No question is a bad question. And I'll kind of tell you why I like what I do as I show you my slides. All right. Give you all a pointer. You can see. Okay, so I'm an analytical chemist, and what that really means is that I use instrumentation. So I use fancy instruments that cost a lot of money to answer some kind of chemical question. More specifically, the instrument that I use is called Mass Spectrometry. So that's what this is. This is a picture of the instrument that I use in my lab that I've done a lot of my PhD work with. And what's really great about Mass Spec is it can kind of sort of tell you how much things weigh. So when you think about if you're on a scale, when you go to the doctor's office, or if you've ever measured something in like a chemistry lab before, it'll give you weight information. Mass Spec does that, but how it does that is it tells you what the mass to the charge ratio is. So everything that exists normally just is neutral, has no positive charges or any negative charges. But what Mass Spec does is you can do what's called ionization. So you can give it a charge. You can add something to it, which will give you a positive charge, or take something off of it, which gives you a negative charge. So that's just like bare bones of what Mass Spec does. But what makes it really great is that we can use native samples. So any sample that you have, like a piece of fruit, a blood sample, anything in between, you can analyze with Mass Spec. It also gives you quantification information. So what the concentration is, how much of something do we have in a given sample? You can find that out with Mass Spec. And it's also really, really fast. A lot of things take, like several hours to do. Some things take days to do. Mass Spec you can do in a few minutes, which makes it really, really great. So my title says using cotton thread to do chemistry. And you might be curious how I even do that, or like why I would even do something like that to do chemistry. Well, this is a picture right here of the two things that I have used throughout my entire PhD work. I used a spool of thread that I bought from the fabric store. So when you think about like if your parents sew or if your grandparents sew, or the thread that they use to play with their fabrics is the same thread that I use to do chemistry. And I also use these really small capillaries. And what that does is it really helps us to do any of these applications that I have outlined here. So thread's really cool because it's widely available. You can get it really anywhere. I think I paid $5 or so for this cotton thread that I've used for the last 5 years. It also has a natural sharp tip because it's really little. So the diameter is really small, so we can take advantage of that to talk about this ionization process that I mentioned before. And it's really easy to use. Like it doesn't take much to use thread to do this level of chemistry. So the things in this awesome graphic that Dr. Sierra Jackson made for us is the things I have circled are all of the things that I have worked on in graduate school. I've looked at drugs. So like if you watch like forensic shows like CSI, NCIS, those types of things, I was able to look at drugs in blood samples, that was my first project. I've also looked at ricinin, which is a small molecule biomarker, and we'll talk about that in a second because this is one of my favorite projects that I've done. And I've also done a lot of what we call omics in the mass spec community. So it really is just, I've looked at really small molecules to tell me something about health and disease. So that was my last project. Sierra, on the other hand, she also did a forensics project and she looked at food. So she's looked at peppers, she's looked at oranges and apples, all with a piece of thread. So it's pretty cool. So you all may or may not have, or your parents may or may not have heard on the news a bunch of times when people say, oh, like they mailed a white powder to a politician. More times than not, that's what's called ricin. And ricin is a toxin that can harm a lot of people. So these are just some clippings of how people have used ricin over the years. So they can send it to President Obama, they've sent it to senators, things like that. People try to ingest ricin, and that's actually really bad for you. You can end up in the hospital if you ingest too much ricin. Now, ricin comes from this plant. So most things have a plant source more times than not. Ricin has a plant source. So this is the castor bean plant, and you might be familiar with castor oil, right? Like you might use castor oil or things like that. You might have castor oil type products in your home. Well, one of the components of the castor bean seed is ricin, which is this really, really large protein. So the good thing about mass spec is that you pretty much can analyze almost anything. I won't say anything because I'm sure there's something that you can't look at with mass spec, but most things you can look at with mass spec. And ricin, which is a really big protein, is something you can look at. The issue with looking at ricin, though, is that it takes forever. And you have to make sure that you have both this red part and this blue part all connected and making sure that they're working properly. And that takes a while to make sure that they're working properly, but also to make sure that they're there. The mass spec that I use is really good for small molecule analysis. So things that only weigh, let's say, between 30 grams to about 4,000 grams, give or take, depending on what their charges are. So because of that, something big like ricin, which is on like the tens of thousands of grams scale, is something that I can't do on my mass spec. My group does small molecule analysis. And instead of taking like 2 days to see if I can look for ricin, I can take 2 minutes and look for these 2 small molecule biomarkers. So ricinin exists in a 5-to-1 ratio. So for every 5 parts ricin, you get 1 part of ricinin. So you can use that as a biomarker. And then this ricinoleic acid is the main component of castor oil, which means in the castor bean seed, it's highly abundant. So we can also use that to tell us if we have ricin in our sample or not. So what I did was I developed a 2-tiered analysis. So the first step was, hey, do we have this ricinoleic acid? Then the second step was, do we have ricinin? So that's what I did using a single piece of thread in 2 minutes, which I think is pretty impressive. So like I mentioned earlier, ricin gets sent in the mail often, and people also try to ingest it. So that means I had to be able to detect it on 2 different substrates. I needed to see if I could see it on an envelope, so on a piece of paper, but also in a biological fluid sample. So when I say biological fluid sample, I mean like blood, urine, things like that, all of those things. So I wanted to see if I could do that. So this is what the workflow looked like essentially. I take an envelope, cut it into little pieces of paper, and then I would put a sample on there. Or if I had a biofluid, I'd cut a piece of thread, dip the thread in the biofluid sample, and then put it in this glass capillary to set it right in front of my instrument. So yeah, so that is all I wanted to tell you all about that. Everything else for this project is super technical, but I would much rather answer any of your questions. These are ways to get in contact with me if you have very technical questions that I can't answer right now. You can shoot me an email. You can follow me on Twitter and shoot me a DM. I'm always on Twitter. Or you can check out my website. So with that, I'll stop sharing my screen and we can just start talking.
Awesome. Thank you so much for sharing that. So our first question comes from Gavin. Could you tell us what exactly a biomarker is?
Yeah, that's a great question. So a lot of times when people are sick, right, your body has different biological pathways that'll release these markers. So if let's say I had a cold, and let's say that in the body, if I get a cold, I'll have an increase of compound A. So you can look for compound A to tell us, hey, okay, this person has a cold. Then maybe another compound, so like compound D is specific to me, maybe like me getting hurt. Those are things that are biomarkers. So we can use those things to tell us if somebody's sick or not.
Awesome. Thank you. Antonetta asks, do the dyes from colored thread interfere with analysis? It's a great question.
So that's a great question. That is one thing we have not looked at hard right now. We've tried to, because we thought that that was going to be an issue. We assumed that because of how thread is made and like the different colors was going to affect analysis. So we use natural colored thread because that would have the least amount of dyes on it, but it probably is going to affect it a little bit.
Very cool. Brielle asks, is there any cotton thread experiments that we can do at home?
Oh, I'd have to think about that. I'll see if I can find a really quick like colorimetric thing that people can do at home. So keep an eye out on my Twitter and on my website for that, but I'll definitely find one for you all to do.
Cool, thanks. Keziah in grade 3 asks, is the thread itself dangerous?
It is not dangerous. So if your parents have like a sewing kit at home, you have that thread, same thread right in your houses right now. So it's not dangerous at all. You can just take it and kind of play with it.
Awesome. Barbara asks, what drew you to this field?
Oh, so analytical chemistry, I originally wanted to do forensics. So, and most forensic scientists are analytical chemists because essentially a big part of their job is characterizing evidence. And the way to do that is through lots of different instrumental techniques. So that's what I do as an analytical chemist. So that's really why I decided to go that route for analytical chemistry. That quickly changed when I got to grad school. I realized that I didn't want to do forensics anymore and I wanted to work more on the health side of science. So thus me switching gears and going to the pharmaceutical industry? That's a great question.
Cool. So when you were working with these like blood samples and these samples that involved like potential like that ricin, were you working with people who were looking for criminals, or were you doing like proof of concept work or like basic science just to understand how things work?
Yeah, so this was all proof of concept. So ricin, the United States has a lot more strict protocols for handling ricin. We can't have actual ricin in the lab. So these experiments were just mimicking if we had ricin, then this is what the protocol would look like. Across the pond, you can kind of have rice in the lab, or like if you worked with a hospital that was emitting ricin patients, you could use their samples. But in the United States, we actually can't do that. So this was just proof of concept based off of what we know about the components of the castor bean seeds.
Very cool. Very cool. So is ricin commonly used because like not, is it more commonly used in this like male like snail male antagonism because it's easy to get through the castor beans, or like why is it the thing that people go to?
Well, it is a little easy to get out of the castor bean seeds, but it also is a white powder. So that could be pretty much anything. It could be sugar, it could be salt, it could be any other white powder. So it's easier to elicit panic that way. So it is also like the panic factor. It doesn't look suspicious because there are a lot of powders we know that are white. So that kind of a catch-22. You can get it out of the castor bean seed if you try hard enough, but it also looks like a lot of other things that can just get sent in the mail regularly.
Totally. Okay. James, grade 3 asks, how long have you been doing this job?
I've been doing this job for 5 years. So, which seems like a very long time after going to high school and then college. But yes, 5 years.
Cool. Nathan, also in grade 3, wants to know how you got to this job. Like what was your path leading you to this?
Yeah, so I went to high school like a lot of people have gone to high school. So like hopefully you all in grade 3 are looking to go to high school. Then after that, you go to a 2-year, 4-year college or institute or university. So I went to school in Pittsburgh, which is in Pennsylvania. And then after that, they were like, hey, you should consider going to graduate school. And graduate school is just more schooling to help you become an expert in something. So I applied right after I finished school in Pittsburgh and got to Ohio State. So I just had really good teachers that kind of helped me get there. So you can definitely ask your teachers to help you get to your next steps too.
Cool. Nelson and Kendall would like to know, why did you have to cut the envelope into small pieces and not just use the whole thing for an experiment?
That is a great question. So why we did that? So the thread is really little, right? Like if you see a piece of a spool thread, I wish I brought mine over here, but thread's really little. And you didn't want to put a full piece of the envelope right in front of the instrument, just in case something happens to it. So a lot of things that people do in science is we do replicates. So if we take one sample, divide it up into smaller pieces, we can keep doing the same experiment just to make sure we have the right answer. If I used it all at once, I wouldn't have been able to tell if I actually got the right answer.
For sure. Cool. Dr. Rylander's science class asked, this is a 5th grade class, my students were curious if there was a way that scientists could stop ricin from hurting you.
Oh, that's a great question. I don't do biochemistry, so I actually don't know. But something that I was reading in a lot of these papers that I read when I was doing this project was that they are trying to find a way to repurpose ricin. So they're trying to see if they could use it to actually be medicine for specific diseases. So I think they're working on that. You all have the right idea.
For sure. Yeah, it's always shocking, like finding the fine line between poison and medicine.
Yes.
Like,
Yes.
it all comes down to dose, and it's wild. It is wild. This is Antoinetta again. What inspired you to continue your study in chemistry as a career specifically?
Yeah, so I really like chemistry. I like being in a lab. I like the fact that with chemistry, things can heat up, change colors, get cold, crash out of solution. I really like that. So it was just a natural progression of me to want to keep doing that because I like being in the lab. And I felt like with chemistry, I could be in the lab a little bit longer than if I had gone a different route. So it's really just because I like it a lot.
That's a solid answer. I like marine biology a lot. That's why I did this. So yeah, very cool. Gavin would like to know, how exactly does mass spectrometry work?
Ooh, okay. So mass spec happens in stages. Okay. So remember when I was saying that in the beginning, when you just have regular things that are regular compounds, things are neutral. So they have no charge. But mass spec, you need a charge to be able to detect it. So there are 3 steps. You first have to give your neutral compounds or your regular compounds a charge. So that's the ionization step. Then after that, you have to sort them out. So if you have a complex mixture, or if I have like a blood sample, it has a lot of stuff in it except that one thing that I want. So we give all of those things a charge, then we have to sort them all out. I need to be able to say that I have this, 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 and this, all in their own order, is increasing weight, essentially. Then after you sort them all out, then you have to detect them. So those are the 3 steps of mass spec: ionization, mass analysis, so that's that sorting process, and detection.
Cool. Awesome. Brielle would like to know, what color is naturally colored thread?
It's white. So it's not anything super interesting. It's just white.
Just white. Because cotton, when it grows, is white. So that makes sense. Very cool. Let's see. All right. So here's a question from Ariel in grade 3. She would like to know whether your experiments have succeeded. So can you talk to us a bit about like what it's like how many of your experiments succeed or fail? Because like I think, when you're in school, pretty much all your science experiments work because they've been tested before. And being a real scientist, we know that's not how it goes. So could you speak to that?
Yes. So being a grad student means that more of your experiments don't work than do work, honestly. So I don't know if I have a ratio, but a lot of my experiments have not worked. Even for this project, I was trying to figure out what I was going to do with being able to look at ricin, and I got stuck because it was really similar to my first paper, and I didn't want to publish the same thing twice. So I really was just trying to figure out other ways to detect ricin in a way that would be interesting to the mass spec community. And I had to talk to my advisor, and I went in his office and was like, look, nothing's working, and I don't know what else we need to do about this. So we kind of put our heads together to come up with our 2-tiered system. But yeah, most of my experiments don't work. The only ones that people like talking about though are are the ones that do work. So that's how we get papers.
For sure. Yeah. The published literature,
Yeah.
that's like your highlight reel. That's not like the day to day, but troubleshooting can be fun and it can be a nightmare. It depends on how long you've been troubleshooting for.
Yes.
Yeah. The number, like I raised squid for my PhD for a lot of the work, and just getting them to survive was like, that was step 0 for the work.
Yeah.
And that took months to figure out. So yeah, science is interesting. Dr. Rylander's science class asks, do emergency services ever have to contact scientists for certain situations?
Yes, and I presented the ricin project because that is the exact scenario where you would have to contact scientists. So if people come and are like, oh my gosh, there's a mysterious white powder that I can't identify, then everything closes. They have to evacuate everybody out, shut everything down, and kind of isolate the mysterious white powder. Then you call your scientists in, and your scientists are going to take the sample and then tell you what exactly that white powder is. So yes, that happens more often than I think people realize.
For sure, yeah. Ethan wants to know, what was your best or favorite experiment and how did you do it?
Ooh, that's a great question. So my first project, I think it was my first project that we were interested in looking at tissues. So like your body's made up of tissues. And sometimes when you go to the doctors or like your parent or guardian goes to the doctor, and they had to get what's called a biopsy. So they're just taking a little piece of tissue from our bodies. And we wanted to see if we could look at tissues with thread. So how we did that was we collaborated with another lab on campus, and they gave me these little squishy beads. So they're like these little purple plump beads that can mimic tissues and how the tissues behave in the body. So I was able to take some thread and a needle, puncture it through the agarose bead, and was able to see what I was actually looking for in the bead. So that, hands down, I think has been my favorite experiment that I've done so far.
That's really cool. So you mentioned that that you have worked with blood and other human liquids over the course of your work. So Karen wants to know, like, did you work with them? How did you work with them? What were you looking for? Like, how did that all come to be?
Yeah, so I have looked at blood and all like blood derived liquids. So plasma and serum come from blood. So I've looked at those. I've also done urine. I've done sweat, done exhaled breath condensate. So if you like blew into something really cold and your breath kind of condenses, so like when it's really cold outside and you blow on a window, I've looked at that too. And all of those, I've looked for something different. So with the blood, I was looking for illicit and pharmaceutical drugs. So sometimes the medicines your parents take, I could look at that in blood. In urine and in serum, I looked for ricinin and ricinoleic acid. So those slides that I showed you. In sweat and the exhaled breath condensate, I'm on a collaboration project with Children's Hospital, and they are interested in looking for biomarkers for cystic fibrosis. So a little bit of everything I think I've looked at so far.
That's wild. How sweaty does a person have to be to collect their sweat?
So you don't get a lot of sweat from people. You get maybe like 100 microliters. So it's not a lot from like, and that's from someone that like sweats a lot. So it's hard to collect that. But that's really it. Like blood, you get like on the milliliter volume, which is great because you can do a lot of stuff with that. But sweat is one of those that's really hard to get very large volumes of.
Yeah, and 100 microliters is like a couple, maybe 2 or 3 raindrops. I'm gonna estimate ish, right? Like,
yeah,
I'm trying to think.
yeah.
So I know what 100 microliters looks like, but it's yeah, it's not a lot. It's like a couple drops basically.
It's not a lot.
A general
Yeah.
drop is like 40 microliters, I think.
Yeah.
There you go. Yeah, I know in the summer you could collect probably a milliliter of sweat off me, but in the winter, forget about it. So, all right, James would like to know, what days do you work and how frequently do you work?
That's a great question. So that's different for every graduate student. I personally only work Monday to Friday because I'm very much against working on the weekends, but that is just very specific to me. Now, I will go in on the occasional Saturday, but Sundays are off limits. I just feel like I shouldn't have to work on a Sunday. So I try to maintain similar hours that your parents work. So, or like your guardians work. So a typical like 9 to 5 is typically my sweet spot of working, but only Monday through Friday.
Beautiful. Yeah, I occasionally would go on the weekends for squid care. But so if you work with animals sometimes, or like cells, sometimes that all gets thrown out the window. But yeah, Monday through Friday is the way to do it. I strongly agree. Let's see. Oh, this is fun. From Ariel. Did you ever get a pink or blue colored liquid in an experiment? And related, Miss Henderson is curious. This is our third grade teacher. If the thread changes colors when you dip it in different liquids.
Yeah, so the thread does change colors. So that's a perk of it being white. So when I was doing blood analysis, if you dip the thread in a little bit of blood, you could kind of watch the blood travel up the thread. So that was kind of cool. If I've gotten, what is blue is sweat. So how they collect for sweat now is they have a little device, like a patch that you wear. And in the patch has this blue dye that kind of helps you estimate how much sweat you actually have. So those samples are actually blue. Blood is red, like we all know it to be. Serum and plasma are like this like yellow, like pale yellow kind of color. Urine is also yellow. But yeah, sweat, that is the one that I didn't realize was gonna come blue. And that's just because of how they collect it, and there's a blue dye there.
So the way you collect the sweat is by having a patch on you, not like following a sweaty person around and micropipetting the sweat.
Yeah,
Okay.
it's like a device that collects it readily. And it has a blue dye. So that's how you can kind of collect it and estimate your volumes.
That's cool. Ms. Ruiz wanted to know how you did that. So I'm glad we got to
Oh,
that.
yes.
Very cool. Dr. Rylander's class says, have you ever made or discovered a new element? What is your favorite thing to do as a chemist?
Oh, so unfortunately, I have never discovered a new element. If I did, I feel like I'd have a Nobel Prize. I think that would warrant that.
Okay.
But I do have a favorite element. My favorite element is called cobalt. And I love cobalt because blue is my favorite color. And one of the complexes it makes is the prettiest shade of blue. So if you type cobalt blue in Google, you'll see the blue that I'm talking about.
It's a very pretty color. You know what's kind of cobalt blue looking? Squid blood.
Really?
Take a look at squid blood. I bet you'll like that color if you like cobalt blue. It's pretty cool. Not to always be bringing it back to school, just
No, you're fine.
connections everywhere. Let's see. Ethan, grade 3, would like to know, how old do you have to be to do your job?
That's a great question. So you can be any age you want to be. I went straight through. So I've been in school since the first day of kindergarten, just to give you all context. And I'm 27, so I've been in school for a very long time.
Yeah.
But I do have classmates that are in graduate school that are a few years older than me, that are 10 years older than me, etc. So you really can be any age you want. As long as you're in the mood to be in school, then you can go to school still.
It's true. Agree. Nathan is curious to know if anything bad has happened to a project you were working
Oh, hmm. I don't think so. I think all of the projects that I've done, I have been able to troubleshoot to some degree. So
on.
I've never had to abandon a project fully because it just wasn't working. But that just says more about me and how I don't let things go than the project itself. So yeah.
Cool. The next question, what is your favorite thing about your job and your least favorite thing about your job?
Okay, great question. So my favorite thing is wholeheartedly being in the lab. So I like being in the lab and creating something new. So no one's ever done thread spray before except my lab. So that's so fun. But that also is my least favorite thing. So it is the same answer. And it's my least favorite thing because you have nowhere else to go. So I had to do all of this research, and my friend Sierra has done all of this research, but that's it. Like we can't talk to other people about it. We can't read about it outside of the things that we published. So that has been a little frustrating, having to try to figure things out kind of on our own without having like textbooks to help us. But it also is really rewarding when I can do a project on something completely different that Sierra did and we both come to the same answer. So that is definitely really rewarding.
Very cool. M. Horner wants to know, are there any other medical uses for thread related to chemistry?
Oh, that's great. So we've really just scratched the surface. So we've only been working on thread spray for as long as Sierra and I've been in grad school. So 6 years total because she graduated last year. And really what we're trying to do now is actually apply it for medical use. So like I mentioned earlier, I'm on a collaboration right now with Children's Hospital to look at cystic fibrosis. We also are trying to extend one of my other projects to look at type 2 diabetes. So we're trying to figure out where the niche is. We also have work in my lab that is looking at malaria. So we're trying to see how far we can take it. Those are just the three off the top of my head that we're working on actively right now.
Very cool. There's a question in here from Nathan. Are chemists doing anything to help with COVID?
That's a great question. So yes, so chemistry is really broad. So I called myself an analytical chemist. So there are these amazing people that are so smart, way smarter than me, called biochemists, and the biochemists are the ones that are helping an awful lot with the COVID-19 response. So they're the ones that are really helping trying to develop the vaccine and really study the virus itself. So a lot of those people are chemists.
Yeah. My buddy Jason, he is a biochemist, and what he's working on is basically like taking what he knows about the structure, like the 3D structure of COVID, and also the drugs that we're trying to use against it, and other things that would interact or like hit the virus. And so it kind of looks like a video game. He does this all on the computer to like see how molecules would stick together or get in the way of each other, all in the body, but knowing what we know about them from like their sequences or their structure from other various stuff. So biochemistry is such a huge field that like what you learn in chemistry in school is just barely scratching the surface, because Jason really never even touches a tube of anything compared to Devin, who's working with tubes and blood and liquids all over the place. So yeah, he's just beep booping and modeling things in 3D space. So lots of ways to be a chemist. All right, next questions. It's from Sasha. This is actually just related to what we just said. Sasha's in grade 8. What are the different types of chemists? Like if we were just to like take a broad brush stroke of all the chemistries, what exists?
Yes. So I'm an analytical chemist. So I'll go in alphabetical order, hopefully. So I'm an analytical chemist. We play with all of the instruments. Then you have your biochemists who study lots of things like in the body. So they use chemistry to really figure out what's going on inside of us. Then we have inorganic chemists. Inorganic chemists play with metals. They're really cool. So they make a lot of materials, and a lot of their things change colors. So I was almost an inorganic chemist just to make stuff that changed colors. You have organic chemists that do something similar. Their stuff doesn't change colors as much though. But organic chemists are the people that are helping make drugs essentially for pharmaceutical companies. You have physical chemists. Physical chemists are the ones, some of them are on the computer a lot, so they do computational chemistry. So similar to what Sarah was just talking about with her friend, like doing stuff on the computer, you can be a physical chemist and do that. I think I hit the 5. Me, biochem, organic, inorganic, physical. Yeah, so those are the 5 main sub-disciplines of chemistry. Oh, you could also do chemistry education. So that's something that's newer. So if you don't necessarily like being in the lab doing the experiments, but you want to help students learn chemistry better, you can do chemistry education work.
Awesome.
So those are the ones.
Great, thanks. Ariel would like to know if any experiment has ever blown up. Have you ever had an experiment just explode?
My research does not have the potential to blow up. So unfortunately, no, I have not had, or I guess fortunately, no, nothing has blown up in my lab.
Sometimes things blow up in biology too. Like if you have bacteria, I've had some things get yucky real quick from bacteria that create gases basically in like who it's a mess. Let's see. So Ariel also wants to know, how hard is it to learn chemistry? Is chemistry, this is a bunch of, and also Dr. Rylander's
Oh.
class, and Gavin, they all are asking, like, is it hard? Is it intimidating?
So chemistry can be really intimidating, but the one thing that I personally like about chemistry is that everything builds on each other. So if you learn something one day, the next day, you're going to use the same thing you learned the day before to do something different. So you always can find an answer that way, because everything is connected. So it's not like you're learning like random facts that somehow fit together. You always can figure out how they fit together based off of something that you learned in a chemistry class. So it is doable. So I don't want you all to think that it's not doable. It's definitely doable. I personally didn't do too hot in organic chemistry when I was in college, but it's okay. I'm about to get a PhD tomorrow, so it's fine. Like it is okay if like one part of chemistry you don't really understand, because it's very broad and very vast, and there's probably a lot of parts of chemistry that you will get and that you'll actually enjoy.
Awesome. Yeah, don't let one class bring you down. Cosign big time on that one. Lisa asks, my students have been studying coral bleaching and learned that the material in coral are used for medical testing. Is mass spectrometry the type of machine that would have been used to understand the makeup and the relationship for the use of humans? So taking like compounds in coral to see if they'd be drugs and testing and all that sort of thing.
It could be. I don't know enough about what they'd be looking for in coral bleaching, but mass spec can be used. It just really depends on the identity of what you're looking for. But there are other instrumental techniques. So maybe if something gives off light or absorbs light, you can do some form of spectroscopy because that measures light. Sometimes if you want another form of characterization, you can use this technique called NMR, which stands for nuclear magnetic resonance. I think that's what it stands for. I don't do NMR, but I'm pretty sure that's the acronym. So NMR also really helps. There are a lot of different techniques, but I'm sure that somewhere down the road, mass spec has been used in some way to kind of help solve that question.
Yeah, totally. So I can actually speak to this a little bit because I don't know anything about chemistry. But when I was in grad school, my buddy Andrea was trying to figure out what compounds inside of bacteria that live inside squid can be used for antibiotics. Because a lot of times bacteria, they're living in environments with lots of other bacteria, and they kind of need to compete with other bacteria to stake their ground. And I think coral do this too with other corals for sure. And so bacteria make compounds that kill other bacteria. And so that's really useful for us when we're trying to kill bacteria that might want to take over our bodies. And so we were biologists, we were squid biologists and microbiologists, and we didn't know really how to do the chemistry part. So my buddy Andrea would do all of these experiments on petri dishes where she looked to see if there were areas on the plate where the bacteria couldn't grow because the other bacteria was kind of like attacking them with chemicals. And so we would take the plate and hand it off to the chemistry folks and say, can you tell us what is in this? And so then they would take that sample and run it through. Now, I don't remember if it was mass spec, HPLC, or what. I think it was a mass spec, and looked for little peaks and said, okay, that thing right there, that is called indigoidine. So that was like the compound that killed the other stuff. And Devin, lots of colors in this stuff. It was the bacteria was dark blue, black, and others were hot pink and orange. And man, it was cool. And so that is how, like, a lot of times biologists and chemists, we all have to work together to answer these big problems, because I only know about squid and my buddy knows a lot about bacteria, and we together with the chemists actually get the answers that humans need to move forward. We can't all do it alone. So thank you, chemistry. All right. This is a fun question from Dr. Rylander's class. As a chemist, have you ever tried to figure out if there is another dimension to our world?
Wow. I have not, but there are chemists that study, there are chemists that do like astrochemistry that probably look at that. So you can be a chemist and you could look at the planets, you could study the moons of the planets, etc. So you can pretty much do a lot of things. on that. But I personally have not done that, but I'm sure that I can find somebody that did.
For sure. That's awesome. It's another case of scientists working together.
Yeah.
Zora would like to know, what's the first project you ever worked on as a scientist?
Wow. Okay. So I also did research as an undergrad. So when I was at the University of Pittsburgh, one of the professors there took me under her wing, gave me to a grad student to do chemistry. So one of the first things that I did with them, they looked at polymers. So most things are polymers. So all a polymer is if you have, units A and B, a polymer is just A and B repeating for however long they repeat for. So that's what a polymer is. So most things, so something like rubber is a polymer. Nylon's also a polymer. Polyester, so stuff your clothes are made out of or like materials in your house, all polymers. So what they did was they made polymers and they actually sequenced A, B, or A, etc. And it was my job to take all of those polymers and characterize them. So I got to learn a bunch of different techniques to figure out what these polymers could do and what we could use them for. So that was one of the first things that I did experiments wise.
Super cool. Awesome. Last question from the audience. Do you only use human blood or do you also use animal blood to do experiments?
That's a great question. I exclusively work with human biological fluids, but my friend Sierra, who I talked about earlier, who also works on third spray, she's looked at canine blood and horse blood for one of her projects.
Very cool. I love the number of times that both of us have talked about like our friends that do related projects.
Yes.
Like, well, I don't do that, but yeah,
Right.
science friends are great. Okay, so we have a couple final questions before we wrap up. The first question is, and we ask these to every scientist who comes on, you have the attention of everyone in the whole world, and you can tell them one thing about chemistry or broadly your area of expertise. What do you tell them?
Oh, that's a great question. I tell them that I can use thread to detect diseases. That would be the one thing that I would start with that and then see what other questions happen after that.
You're gonna have their attention after that because that's a wild thing to say. So that's
I love a good pitch. Yeah, I love pitches for all my projects.
solid hook. And the second question is, you still have everybody's attention in the world, and you can tell them one thing about literally anything, one message to get across to all humans. It can be as big picture important or silly and insignificant as you'd like. What do you tell all the people of the world?
All the people of the world. I would tell all the people of the world that it does not hurt to be kind. I think the reason why I've been so successful and where I am today is strictly because I'm a nice person. So always be kind, friends.
Solid. I co-sign that as well. That's awesome. Is there anything that you'd like to plug? Anything you'd like us to know about? Any other information? This is your time to tell us literally whatever you want.
Yeah, so I wear a lot of hats. But one of the things that I think is really cool is if people are looking for other really awesome Black chemists to read about or to talk to, you can search the hashtag Black in Chem, so short for Black in Chemistry, on Twitter. That is also the website link, so it's blackinchem.com, and also our Twitter handle at Black in Chem. So that is like a one-stop shopping for all things related to Black people doing really cool chemistry. I also have a blog that's on my website that's called Max Scientists, and that is where I'm super candid about what it's like being a grad student and what my journey has been like so far. So those are two things that I would plug.
Awesome. And where can we find you on social media?
On social media, I am at Devin, so my name, how it's spelled, Devin_11, but 11's all spelled out in the how you spell 11. So that's where I am on Twitter.
Very cool. And you can find all that on the Skype a Scientist Twitter. We've been linking all week, so you can just click that and easily find that Twitter account. Awesome. Well, thank you so much for answering rapid fire questions for 40 minutes straight. We really appreciate it. Thanks for sharing your knowledge with us, your perspective. We really appreciate the time that you took to spend with us. Erin, thank you so much for signing, as always. Anything else before we wrap up?
No, don't have anything. Just thank you all for letting me come talk to you and answer your questions.
Awesome. Thank you. We hope to see you all on Wednesday to talk about the ocean at 1 p.m. Eastern. You can find out all the information at skypeascientist.com. Also, Skype a Scientist is a nonprofit organization, and we rely on small donations to keep our ship afloat. If you can support us at patreon.com/skypeascientist, that would be super appreciated. Just $3 a month even is huge for us. So if you can do that, we would really, really appreciate it. We'll see you all on Wednesday. And thank you again, Devin and Erin, for spending time with us. Bye, everyone.
Bye.